Hi guys, Miss Francis here today to talk to you about the Calvin cycle. Before we get to the Calvin cycle, now that we're finished with the light reactions, let's talk about other names that scientists have for the light reactions. The light reactions can be called the light-dependent reactions because the light is needed to excite the electrons and begin the process. It's also called photolysis because the water is split or lysed using photons of light, but that only happens in non-cyclic photophosphorylation. It's also called photophosphorylation. Why? Because light is the initiator of the reactions, which add a phosphate back to ADP, so ADP gets phosphorylated in order to make ATP. And finally, the light reactions are sometimes referred to as the photochemical reactions. So now let's explore the Calvin cycle. So in a nutshell, the Calvin cycle regenerates its starting material after the molecules enter and leave the cycle. Carbon dioxide enters the cycle and leaves as sugar, but the sugar that leaves the Calvin cycle isn't glucose, but a 3-carbon sugar called G3P for short, or glyceraldehyde 3-phosphate. Now this cycle, the Calvin cycle, depends on the energy of ATP and the reducing power of NADPH that we got when we did those light reactions. So just like the light reactions have different names, scientists have different names for the dark reactions. The dark reactions can also be called the Calvin cycle because the scientist Melvin Calvin discovered the pathway of carbon using carbon-14 as a tracer within these reactions. It can be called the light-independent reaction because light isn't needed. The reactions can occur in the day or in the night, unlike the light-dependent reactions where light is needed. It's also known as the carbon-fixing reaction because the carbon dioxide is fixed or made into sugar. So there are 3 phases in the Calvin cycle. We're going to annotate an image, then fill in our note packet, and hopefully by the end of this video, you'll understand the Calvin cycle. So the first phase is the carbon fixation stage. Well, where does this carbon come from? I get carbon dioxide, which is a 1-carbon sugar that enters through the stomates of a leaf. Now, speaking of the leaf, where exactly does the Calvin cycle occur? The Calvin cycle takes place in the stroma of the chloroplast that's found inside of the green plant. All right, so we've got this 1-carbon sugar, carbon dioxide. This 1-carbon sugar is going to combine with a 5-carbon sugar called ribulose bisphosphate, also summarized as REBP. The enzyme Ribisco is going to be used to combine carbon dioxide and ribulose bisphosphate into a 6-carbon sugar. But that 6-carbon sugar is extremely unstable, so it immediately splits into 6 3-carbon sugars called phosphoglycerate. So at this point, you might be wondering, well, wait, how did I get 6 3-carbon compounds? I started out with 3 1-carbon compounds, right? 3 carbon dioxides, and I added 3 5-carbon compounds, so 3 5-carbon compounds. When I combined my first carbon dioxide with ribulose bisphosphate, I made a 6-carbon sugar, right? Because I've got 3 of carbon dioxide and 3 of ribulose bisphosphate. When I combined my second carbon dioxide with my 5-carbon ribulose bisphosphate, I made a second 6-carbon molecule. When I combined my third carbon dioxide with my third ribulose bisphosphate, I made my third 6-carbon molecule. But they were very unstable, so all of those molecules split, and I got 1, 2, 3, 4, 5, 6 phosphoglycerate. So that's how I ended up with 6 phosphoglycerate after the carbon fixation phase of the Calvin cycle. So let's fill out our note packet. So in our note packet, it says, what is the starting reactant? Well, the starting reactant is ribulose bisphosphate, which is a 5-carbon sugar. Which is a 5-carbon molecule. What is the reaction that's going to occur? Well, the reaction is that my ribulose bisphosphate is going to combine with carbon dioxide. And how does it combine with carbon dioxide? It uses an enzyme, Ribisco, in order to perform that reaction. Now remember, carbon dioxide is a 1-carbon compound. So I end up with a 6-carbon compound that is extremely unstable. It's extremely unstable, so it immediately splits into a 3-carbon molecule known as PGA. The next stage or phase of the carbon cycle is the reduction phase. There's a lot written on here, so just stay with me. When we did the carbon fixation stage, we ended up with 6 3-carbon molecules called phosphoglycerate. That phosphoglycerate is going to get a phosphate added to it, forming bisphosphoglycerate. Where did that phosphate come from? It came from the ATP that we made during the light reactions. That bisphosphoglycerate is going to get reduced because NADPH will be oxidized. So NADPH is going to lose electrons, lose hydrogens, and bisphosphoglycerate is going to gain those electrons and be converted into our first sugar. Our first sugar is called G3P, and we're going to make 6 of these G3Ps. Where do these 6 G3Ps go? Well, 1 G3P is going to be used by the plant to form glucose and other organic compounds, but 5 of the G3Ps are going to be recycled so that I can continue this cycle of making glucose. So let's fill out our note packet. So it says, what molecules get used from the light reaction during this phase? Well, ATP and NADPH from the light reaction. What products are formed and how many? You get G3P and you get 6 of them. And this is our first sugar. So that the final product of the Calvin cycle is not glucose, it's actually this G3P. What becomes of these 6 G3Ps, which is also called pGal? One of them is going to be used to make glucose, so it's going to be used as an output, but 5 of them are going to be recycled into the Calvin cycle. Those 5 3-carbon sugars are going to be rearranged into 3 5-carbon sugars, those ribulose bisphosphate that we started out with. So that's the next phase. So this third and final phase of the Calvin cycle is called regeneration, because we are going to regenerate that ribulose bisphosphate, which is the carbon dioxide acceptor. So what happens is 5 3-carbon sugars, right? So I've got 1, 2, 3, 4, 5. And what are those 5 3-carbon sugars? Those are your G3Ps. Those 5 G3Ps, with the help of ATP from the light reaction, are going to be converted into 3 5-carbon sugars. What are those 3 5-carbon sugars? Well, that's your ribulose bisphosphate that's needed for this cycle to even occur. So let's fill out the note packet, and then I want to go back and point something out. So what molecule is regenerated and why? My ribulose bisphosphate is regenerated. Why? Because it's a carbon dioxide acceptor, which is needed for this whole reaction to occur. How is this phase fueled? By the ATP that came from the light reactions, and then how many G3P make how many Rubisco molecules. So 5 G3P make 3 ribulose bisphosphate. So I just want to point something out. There are 3 carbon dioxides used during the Calvin cycle. That means this cycle is going to spin 3 times. But that only gives me 1 G3P, which is a 3-carbon sugar. Remember that glucose is a 6-carbon sugar. So in order to get those 6 carbons, I would need 2 G3Ps. In order to get those 2 G3Ps, I have to do this 2 times, which means that 6 spins is how I'm going to make my glucose. Okay, so what do you really need to know about the Calvin cycle? That 6 carbon dioxide molecules turn the cycle 6 times. And from those 6 spins, or those 6 turns, I get 2 molecules of G3P, of P-gal, which is our first sugar. Those 2 molecules of G3P then can be made into a 6-carbon glucose molecule. You need to know that GP3 is the first stable sugar made, since G3P is a 3-carbon sugar. You also need to know, like we mentioned earlier, that for every 2 G3Ps, I'm going to get 1 glucose, and then those glucoses can eventually form starch and other polysaccharides. Calvin is credited with discovering the pathway that carbon takes during photosynthesis. It was called the lollipop experiment because the setup kind of looks like a lollipop. So what he did was he exposed algae to carbon dioxide that was radioactively labeled with carbon-14. And then at intermediate steps throughout photosynthesis, he identified those carbon compounds using chromatography and thus was able to trace the pathway of carbon throughout photosynthesis. So let's trace the atoms in photosynthesis. So if I'm looking at carbon dioxide is ultimately going to get reduced, meaning I'm going to gain electrons, it's going to gain hydrogens. During the Calvin cycle, the carbon and oxygen were rearranged to ultimately end up in glucose. Hydrolysis is going to occur. So my water is going to split. When my water splits, I get my oxygen. And then through the help of NADPH, which came from the light reaction, I am able to help form glucose.